Carbohydrate chemistry. Under this, we have monosaccharide, oligosaccharide, and polysaccharide. The first one is monosaccharide. Monosaccharide are also called as simple sugars. They are soluble in water and they are sweet to taste. They consist of single polyhydroxyaldehyde or ketone. They are classified based on the number of carbon atom and the type of functional group. For example, 3 carbon is triose, 4 carbon is tetrose, 5 carbon is pentose, and 6 carbon is hexose. The most abundant monosaccharide is D-glucose. Other examples of monosaccharides include glucose, fructose, and galactose. Functions of monosaccharide include production and storage of energy. The second one is oligosaccharides. These consist of 2 to 10 monosaccharide units and are linked by glycosidic bond. Disaccharides are the most abundant. Disaccharides are 2 monosaccharide units linked by glycosidic bond. Sucrose formed by glucose and fructose, lactose from glucose and galactose, and maltose, glucose. Sucrose is a non-reducing sugar. Lactose and maltose are reducing sugars. Why is sucrose non-reducing? Because both the anomeric carbon are involved in the glycosidic bond. The first one is sucrose, which breaks down to form glucose and fructose. It is formed by plants and it is a product of photosynthesis. It does not contain free anomeric carbon atom as it is involved in glycosidic bond, hence it is a non-reducing sugar. The enzyme involved is invertase. The second one is lactose, which breaks down to form glucose and galactose. It is a reducing sugar. It is present in milk. The enzyme involved is lactase in human beings, and the enzyme involved in bacteria is beta-D-galactosidase. The third one is maltose. It is produced by the digestion of starch and glycogen. The enzyme involved is alpha amylase. Now let's see polysaccharides, which are also called as glycans. These are formed by 10 or more monosaccharide units. Polysaccharides are further divided into homopolysaccharide, which are called as homoglycans, and heteropolysaccharides called as heteroglycans. Let's see the differences between homopolysaccharide and heteropolysaccharide. Homopolysaccharides are made up of same type of monosaccharide units, and heteropolysaccharides are made up of different types of monosaccharide units. Examples of homopolysaccharide include starch. Under this we have amylose and amylopectin. Amylose is unbranched and it is soluble. Amylopectin is branched. The second one is dextrin. Partial hydrolysis of starch by an acid produces dextrin. The third one is glycogen, which is also called as animal starch. It helps in storage of blood glucose and maintenance of blood glucose. The fifth one is inulin, which is hydrolyzed by inulinase. It is a polymer of D-fructose and it is used for GFR. It is metabolically inert. The sixth one is dextran. It has alpha-1,6 linkage. It is produced by yeast or bacteria and it is used as a drug, which is a plasma expander. The fourth one is cellulose. It is an unbranched polymer. The humans lack the enzyme cellulase and therefore it cannot be digested or absorbed. Now let's see the example of heteropolysaccharides. The first one is agar. It is obtained from seaweeds. It is non-digestible. It helps in the treatment of constipation and it is made up of galactose unit. The second one is gum and pectin. These are also called as non-cellulose fibers. They decrease the cholesterol. They are used for diabetic patients as it reduces the increased risk of blood glucose. The fourth one is mucopolysaccharides, also called as GAGs. These were first isolated from mucin, which led to the name mucopolysaccharides. GAG is unbranched, made up of repeating disaccharides. GAG comprises of amino sugar and uronic acid. The GAG is attached to core protein to form proteoglycans. This resembles a bottle brush structure. The core protein, that is the proteoglycan monomer, binds with high hyaluronic acid by an ionic bond forming proteoglycan and is aggregated by link protein. Proteoglycans comprise maximum of carbohydrates. High hyaluronan is a long chain of repeating disaccharide. It does not contain sulfate group. It does not attach directly to the protein whole. GAGs are found in synovial fluid of the joints, bones, cartilage, and the vitreous humor of the eye. Now let's see the type of GAGs, the location and function. The first one is chondroitin sulfate, present in tendons, cartilage, and cornea, helps in compressibility of the cartilage in the weight-bearing joint. The second one is keratin sulfate, present in the cornea, helps in the transparency of cornea. The third one is dermatan sulfate, present in skin and blood vessels, helps in the transparency of cornea. The fourth one is heparin sulfate, present in the skin and fibroblast, acts as receptors in the cell membrane. The fifth one is heparin, present in the liver and lungs, helps in anticoagulation and lipid metabolism. The sixth one is high hyaluronic acid, present in the joints and vitreous humor of the eye, helps in lubrication and healing of wound. Function of GAGs. It is called as the ground substance or the cementing substance as it forms the extracellular matrix. They have negative charge and bind with large amount of water which protects against shocks. They act as molecular sieves. They provide elasticity to cartilage or substances. That is, when the GAG is compressed, water is squeezed out and the GAG becomes smaller. After release of compression, it regains the original volume. GAGs also help in the lubrication of joints and the enzyme involved is high hyaluronidase. Glycoproteins. These are proteins to which oligosaccharides are covalently attached to the polypeptide chain. They are branched and they do not contain repeating disaccharides. Albumin is not a glycoprotein. Glycoproteins are made up of 8 types of sugars. Functions of glycoproteins. Except albumin, all the plasma proteins are glycoproteins. They act as integral proteins, antibodies, hormones, and coagulation factors. They are used as a lubricant and protective agent. They regulate the lifespan of proteins and they serve as cell-to-cell interaction. They prevent denaturation and also help in solubility. Glycocomics. Glycocomics are systematic characterization of all carbohydrate components of a cell, including those attached to proteins or lipids. Glycocomics provide new information on the roles of sugar in health and disease. It indicates the target for a drug. And carbohydrate component of a cell depends on mass spectrometry.